In this lecture, we are going to discuss two extracellular appendages of the bacterial cell, flagella and pili or fimbriae. Let us begin with the first extracellular appendage of bacterial cell, the flagella. It is the locomotory organ of bacteria. They move by means of flagella, which are thin, hair-like appendages extending out from the cell wall. They differ from the eukaryotic flagella in being thinner and simpler in structure. The number and location of flagella serve as a criterion for classification of bacteria. The number ranges from 1 to several hundred. If the bacteria have flagella at the ends, it is called polar flagella. And if it has the flagella along the sides, it is called lateral flagella. Let us now discuss the different arrangements of bacterial flagella. Bacteria may have only one flagellum at one end and are called monotrichous. Example, Pseudomonas aeruginosa. If the bacterium carries a single tuft of flagella, it is said to be lophotrichous. Example, Pseudomonas fluorescens. When flagella appear at both ends of the cell, either single or as a tuft, the bacterium is amphitrichous. Amphi in Greek means at each end. Example, Aquaspirillum serpens. Bacteria that are covered all over in flagella are said to be peritrichous. Example, Salmonella typhi. Bacteria without flagella are called atrichous. Let us now discuss the ultrastructure of flagella. A bacterial flagellum has three basic parts: a filament, a hook, and a basal body. The filament is the rigid helical structure that extends from the cell surface. The helix may be right-handed, as in the case of Caulobacter crescentus, or left-handed, as in the case of Salmonella species. It is composed of the protein flagellin, the molecular weight of which is 30,000 to 60,000, and is a hollow tube 20 nanometers thick. The primary structure of flagellin is such that the amino acid sequence of both terminal regions is well conserved, whereas that of the central region is variable even among species of same genera. This hypervariability gives rise to hundreds of serotypes of Salmonella species. During synthesis of the filament, flagellin molecules coming off of the ribosomes are thought to be transported through the hollow core of the filament, where they attach to the growing tip of the filament, causing it to lengthen. This type of synthesis is called self-assembly. Thus, the flagellum grows at its tip rather than its base. The filament ends in a capping protein. The cap protein forms a star-shaped pentamer and is responsible for the self-assembly of filament. In its absence, the flagellin is secreted in the medium as monomers and fails to polymerize. The hook, as the name suggests, is sharply curved almost at right angle to that of the filament. It connects the filament to the basal body, causing it to point directly away from the cell. It is shorter, almost 55 nanometers long. The hook is made up of a single kind of protein called hook protein or FLGE. The hook does not self-assemble like that of the filament but requires a helper protein FLGD. There are two minor proteins between the hook and the filament called hook associated proteins HAP1 and HAP3. The basal body anchors the flagellum to the cell wall and the cytoplasmic membrane and consists of a rod and a series of rings. Gram-negative organisms have 4 rings. L ring associates with the lipopolysaccharides of the outer membrane. P ring associates with the peptidoglycan layer. M ring is embedded in the plasma membrane and S ring is directly attached to the plasma membrane. The L and P rings bind together to form the LP ring complex, and this complex serves as a rigid bushing to support the rod where it is joined to the hook of the filament on the cell surface, which holds the entire flagellar structure stably. The LP ring complex is resistant to extremes of pH and temperature. The MS ring structure is regarded as the construction base onto which the other flagellar substructures attach. The rod is made up of 4 proteins and is the site where the P ring attaches. Gram-positive organisms have only 2 basal body rings, P in the peptidoglycan layer and M in the plasma membrane. We have just seen that the flagella is made of filament, hook and basal body. Let us understand the structure of the basal body slightly more in detail. The basal body acts as a molecular motor which enables the flagellum to rotate and propel the bacterium through the surrounding medium. In fact, the flagellar motor rotates very rapidly. The bacterial flagellum is driven by a rotary engine composed of proteins MOT-A and MOT-B located at the flagellum's anchor point on the inner cell membrane. Obviously, for the motor to rotate, energy is required, which is provided by proton motive force, that is, by the flow of protons across the bacterial cell membrane due to a concentration gradient set up by the cell's metabolism. The rotor transports protons across the membrane and is in turn turned on in this process. Thus, the proton motive force is used to spin the hook and the filament, driving the bacterium forward. The rotor by itself can operate at 6000 to 17,000 rpm, but with a filament attached, usually only reaches 200 to 1000 rpm. Bacteria moves through the action of the flagella. Well, the bacterial motility is not aimless but is governed by the process called chemotaxis, the directed motion of bacteria towards or away from positive or negative stimuli. When bacteria move towards attractants, it is called positive chemotaxis, and when they move away from the repellents, it is called negative chemotoxis. Examples of attractants are nutrients like sugars, amino acids, peptides or growth factors, whereas that of repellents are acids which cause changes in pH and alcohols. In response to positive stimulus, the bacteria runs forward, and in response to negative stimulus, it tumbles. In the case of monotrichous bacteria, the flagella rotates counterclockwise, causing the bacterium to move forward with the flagellum trailing behind it in response to positive chemotactic stimuli. This forward movement is termed the run. Negative chemotaxis causes clockwise rotation of the flagellum and results in a random tumbling motion. Peritrichous bacteria move in a similar fashion, even though the situation is somewhat complicated by a requirement for bundling of the flagella to produce coordinated action during counterclockwise rotation. The tumble in peritrichous bacteria is the result of bundle disruption during clockwise flagellar rotation. During a run that lasts about 1 second, the bacterium moves 10 to 20 times its length before it stops. In the case of a tumble, the movement lasts only about 1/10 of a second and no real forward progress is made. Let us understand the forward run and tumble by seeing this short video. In plain medium containing no attractant or repellent, the length of runs is random and the bacteria move about aimlessly in the solution, as can be seen in this clip. Thus, bacteria respond differently in response to a stimulus, whether it be an attractant or a repellent, and show a random response in absence of either. Since motility is the primary criterion for the identification of bacteria, several techniques have been developed to demonstrate bacterial motility directly or indirectly. Direct methods involve looking for the presence of flagella by staining. Flagella can be coated with dyes like pararosaline or basic friction along with a mordant like tannic acid. The binding of the dye adds extra width to the otherwise very thin flagella, allowing them to absorb light, making them visible. If a bacterium possesses flagella, it is presumed to be motile. Indirect methods involve looking for movement. Microscopy is a very commonly used method. It is possible to watch living bacteria swim around using the light or phase contrast microscope. For this, a hanging drop preparation is made. You can start by taking a clean cavity slide and a cover slip. Then place a drop of broth culture in the center of the cover slip. At the four corners, place small drops of petroleum jelly. Now, invert the cavity slide on the cover slip with cavity facing downwards so that the drop hangs in the cavity. Now observe the slide under the microscope. One must look for transient movement of swimming bacteria. Most bacteria, because of their small size, will shake back and forth in a wet mount observed at 400x or 1000x. This is Brownian movement due to random collisions between water molecules and bacterial cells. True motility is confirmed by observing the bacterium swim from one end of the microscopic field to the other side as observed in the video in the previous slide. The second method is using motility medium. This is a semi-solid medium that will hold non-motile bacteria in place, but motile microbes can swim through it. The medium is inoculated with the bacteria in a straight line stab with an inoculating needle. After incubation, if turbidity due to bacterial growth is observed only near the stab line, then the bacterial culture is non-motile, as can be seen here. But if the turbidity indicating bacterial growth is seen away from the line of the stab, it is evident that the bacteria were able to swim through the medium, as can be seen in this picture. Let us now talk about the second extracellular appendage, pili. A pilus, plural pili, or fimbrium, plural fimbriae, is another extracellular appendage. It is a thin hair-like structure on the surface of a cell, especially in gram-negative bacteria, but is also found in some archaea and gram-positive bacteria. They are similar to flagella in being extracellular appendages, but differ from them in being thinner. Pili were first observed under electron microscope by Dewey in 1955. He found these non-flagellar filamentous appendages and correlated their presence with the ability of E. coli to agglutinate red blood cells. He called them fimbriae. 10 years later, Brinton observed fibrous structures responsible for conjugative transfer of genetic material in bacteria and named them pilus. Since then, the terms pilus and fimbriae are used interchangeably for all types of non-flagellar filamentous appendages. Pili are around 2 to 11 nanometers in diameter and up to 20 millimeter in length. They are composed of a single type of protein subunits called pilin or fimbrillin, the molecular weight of which is 17,000, and these proteins are arranged helically. Fimbriae are either located at the poles of a cell or are evenly spread over its entire surface. Let us now discuss the functions of pili. Pili play an important role in attachment and adhesion by helping the cell to bind to solid surface. Because of this characteristic, pili are very important in pathogens and act as virulence factors. Often, if a pathogen loses its ability to produce pili, it also loses its ability to cause disease. Pili are also important in producing biofilms, which are the slimy layers covering our teeth, tongue, the bottoms of ship, trickling sewage, treatment plants, and the rocks in lakes and streams. Sex pili are longer, hollow protein rods that are mainly found on gram-negative cells. They are required for bacterial conjugation, whereby there is a transfer of genetic material between bacteria through direct physical contact. The bacterial cell which harbors the pili is called the donor cell and it connects with the recipient cell via the pili to form the conjugation tube between the two bacterial cells. Once the two cells are in contact and the mating pair has been established, there is transfer of plasmid DNA from the donor to the recipient. The exchanged plasmid can add new functions to a bacterium like antibiotic resistance or metal tolerance. We will discuss more about conjugation when we deal with the topic of plasmids in bacteria.